Okay, so we're going to start talking about visualization of structures at the cellular level and down to the level of individual organelles as well. Um, Remember, way back on day one in this class, we talked about the definition of what does visualization even mean, specifically with reference to scientific visualization. And I gave you that quote from the Association of Medical Illustrators that medical illustrators draw what can't be seen, watch what's never been done, and tell thousands about it without saying a word. And the first part of that is, we draw what can't be seen. So a big part of what we do is to make the invisible visible, to see things that are too small to be seen with the naked eye, and to make these things visible for people to see. So I want to talk a little bit about exactly how small is small. How small are we talking about? How small does something have to be before we can't see it? And then focus the rest of the lecture on what kind of technologies are available to help us magnify things so that we can see some of these very small structures. So I want to start off with a little refresher about scale, relative size, and about units of measurement. I'm hoping that this is all review for all of you. Hopefully you've all had this in a science class in middle school or high school or biology class. If any of you have taken some chemistry, they may deal with some of this stuff too. I know here in the US, we don't work in the metric system very often, so this may require a little bit of a refresher about some of these units. In in the world of science, the metric system is king. So even though, as Americans, we're used to using the old British imperial system of inches and feet and miles and pounds and things like that, in the sciences, everybody uses metric system. Uh, so looking at this little chart that shows some common units of measurement that are used in the sciences, hopefully the most familiar to you is probably the meter. Even if you don't know exactly how big a meter is, hopefully you have some general sense for roughly how big it is. It's a little over 3 feet, so it's a little bit bigger than a yard. Those of you who are football fans, you have a sense for what a yard is. The Buffalo Bills couldn't get 1 yard in their playoff game, unfortunately. Um, so it's like 39 inches or something like that is the meter. Um, so, the size of a small child. I mean, I'm not good with kids' ages. Maybe, I don't know, typical 6, 7-year-old kid, maybe about 3 feet high, thereabouts. So something in that scale of, like a small human. And the meter is represented simply by the letter M. That's the symbol that's used when we're talking about meters. Hopefully you are also familiar with scientific notation. Again, you might need a little bit of a refresher, but you should, and anybody who's a medical illustrator should be very comfortable with scientific notion. 3.6 times 10 to the 3, you should just instinctively know exactly what that means. What does 10 to the 3 mean? Or in this case, what is 10 to the minus 2, 10 to the minus 3? So whenever you see the minus sign in the exponent, it actually means 1 over, 1 divided by. So the next common unit of measurement in the metric system is the centimeter, which is 10 to the minus 2 meters, or to put it a different way, 1 divided by 10 to the 2 meters, and 10 to the 2, 10 squared is 100. So a centimeter is 1/100th of a meter, represented by the symbol cm. And, I put width of a pencil in the chart. That may be kind of a fat pencil. Centimeters a little bit less than a half an inch, but something, on that scale, half inch thereabouts. The next common unit of measure is the millimeter in the metric system. This is 10 to the minus 3 meters. So 1 divided by 10 to the third, or 1/1000th of a meter, 1 divided by a 1 with 3 zeros after it. So it's 1/1000th of a meter, or another way of putting it is it's 1/10th of a centimeter. Millimeters represented by the mm symbol. And, I put here that's the size of an ant, although ants are pretty variable in size. Not those great big carpenter ants. I mean, the big ants are like a whole centimeter long, if not more. I'm talking about the little teeny tiny ants, or the head of a pin. In fact, even a head of a pin may be a little bit bigger than a millimeter, but something on that scale. You know, head of a pin, very tiny insect, something like that. Now, if you look down at the bottom, um, the human eye can see things down to about 10 to the minus 4 meters. So that would be 1/10,000th of a meter. Or another way of putting it is it'd be 1.1 millimeters or 1/10 of a millimeter, or 1/100th of a centimeter. There's a lot of different ways of putting it. So, you know, 1/10 of a millimeter. So imagine something the size of a tiny ant or something the size of a head of a pin, and 1/10 of that. Now you're getting pretty darn small. And 10 to the minus 4 meters or 1/10 of a millimeter, that's about the thickness of a human hair. And that's about the smallest thing that a human can make out, distinguish with the naked eye. Anything smaller than that is going to require some form of magnification to see it. So then we get down into really small things, some very small units of measure that may not be quite as familiar to you, but these are definitely things you should brush up on and be familiar with throughout your career as a medical illustrator. If you do anything at the cellular or molecular scale, you're going to need to know these other units of measure. So the next smaller unit is the micrometer, or more commonly referred to as simply a micron. And that is 10 to the minus 6 meters. So 1/10 with 6 zeros after it. So that's 1/1,000,000th of a meter, or another way of putting it is it's 1/1000th of a millimeter. So imagine that head of a pin divided into 1,000 little subunits. The micrometer or micron is represented by the Greek letter mu, followed by the regular letter m. And now we're down to the cellular scale. If I remember correctly, a typical red blood cell is about 6 microns in diameter. Other cells in your body are going to be somewhere in that same, you know, roughly in that same range of a few microns up to, you know, maybe 10 microns for some of the larger cells in your body. Continuing to get much smaller. The nanometer is 10 to the minus 9 meters. So that would be 1/1 with 9 zeros after it. That's a billion. So this is 1/1,000,000th of a meter, or 1/1000th of a micron. So 1,000 times smaller even than a micron. The nanometer is represented by the nm symbol. And now we're down to molecular scale, at least some of the larger molecules like DNA, which is a pretty big molecule. The DNA double helix is about 20 nanometers in diameter. So we're down to that kind of scale. And then even smaller than that, a very tiny unit, probably the tiniest unit of measurement that you'll ever encounter, at least in the sciences, in the natural sciences, is the angstrom. This is 10 to the minus 10 meters, or 1/10,000,000th of a meter, 1/10th the size of a nanometer. And the angstrom gets its own weird symbol. I forget how you even type this on the keyboard, but you have to hit like Option A or Option Shift A or something like that to get the angstrom symbol. It's a capital A with a little circle over the top of it. And now we're down to the size of individual atoms when you get down to angstrom level. So the other thing that's shown on here relative to these objects of different sizes and these different units of measure is an image over at the far right that shows the wavelengths of different forms of electromagnetic radiation. That's what the ER is short, electromagnetic radiation. Electromagnetic radiation is a form of energy that travels in waves. And it includes microwaves that you use, you know, to heat up your frozen dinners, radio waves, television broadcast signals that travel through the air. And then different forms of light are also different forms of electromagnetic radiation. So there's infrared light, which has longer wavelengths than visible light. You can't see infrared light. Visible light is a form of electromagnetic radiation that has a very specific wavelength. And I tried to get this chart of wavelengths to actually match up with our scale over there. Things like microwaves, believe it or not, have very long wavelengths. They're literally measured like in meters. Radio waves and TV broadcast have wavelengths, in the millimeter range, somewhere in there. And then when you get down to visible light has wavelengths that are about 10 to the minus 7 meters in length. So 1/10 of a micron is the length of these individual waves in visible light, or another way to put it would be, 100 nanometers. That's all, in the range of 10 to the 7. In fact, very specifically, visible light ranges from about 400 nanometers, or 4 times 10 to the minus 7, to about 700 nanometers, or 7 times 10 to the minus 7 meters in length. And then there are other forms of electromagnetic radiation that have even shorter wavelengths, ridiculously tiny wavelengths. The electron, believe it or not, even though we think of the electron as a particle, it has this, what they call the wave-particle duality. It's a quantum mechanics thing, that it sort of functions like a particle, but sort of functions like a wave. So we've described electrons as having a wavelength that are down around 10 to the minus 10 meters, so 10 to the minus 9 to minus 10. And then X-rays have even smaller wavelengths, down to 10 to the minus 10, 10 to the minus 11 meters. There's even something with a wavelength smaller than that, something called gamma rays, which is a type of cosmic radiation that has even shorter wavelengths. That is going to be important for a couple of reasons. This whole thing about wavelength is important for 2 reasons. I mentioned that visible light has a wavelength somewhere in the range of 10 to the minus 7 meters. It's like anywhere from 4 times 10 to the minus 7 to 7 times 10 to the minus 7 meters. An important principle in visualizing anything is that electromagnetic radiation can only reflect off of something that's bigger than its own wavelength. So that means that visible light can only reflect off of objects that are larger than 10 to the minus 7 meters in size. If you have an object that's smaller than that, like a molecule or an atom, visible light cannot reflect off it. Therefore, that means, and in order to see something with visible light, the light has to hit the object and bounce back to your eye. If the light can't bounce off of something that's too small, that means that it is theoretically impossible to see it. So individual atoms and molecules, because they're smaller than 10 to the minus 7 meters in size, it means that light cannot reflect off of them. Therefore, it is theoretically impossible to see anything that's smaller than the wavelength of visible light. So it's you can't see anything smaller than 10 to the minus 7 meters, even with all kinds of magnification. Just literally the light won't bounce off of it, so it can't be seen. Yes, question.
Does that mean like stop?
It just passes right by it. Yep. And doesn't reflect off of it at all. So we're going to talk about light microscopy, and I'm going to mention that one of the shortcomings of light microscopy is the fact that it can't see anything smaller than 10 to the minus 7 meters, no matter how good the microscope is or how powerful it is. That is the theoretical limit of what can be seen with visible light. Now, also a little bit later, we'll talk about ways that you can see things that are smaller, but you have to use electromagnetic radiation that has smaller wavelengths, like electrons. That's the principle behind electron microscopy. Or X-rays. There's something called X-ray crystallography that we'll talk about a lot more later in the semester. That's how you're able to see like individual atoms and individual molecules, because X-rays have this very, very tiny wavelengths. Right, so let's talk about different ways of magnifying things so that we can see some of these very, very tiny objects. We'll start with light microscopy, which is the oldest of these technologies that allows us to see things that are pretty small. The light microscope has been around since the 1500s, and the earliest light microscopes used just a single lens. In fact, there was one developed by a guy named Anton van Leeuwenhoek that just used a droplet of water and the curvature of the droplet of water acted like a lens to magnify very small things. But a lot of lenses were ground out of glass, creating sort of a round or lens-shaped object out of glass. And then the real big advance in light microscopy came with the development of the compound microscope that uses two different lenses. There's an ocular lens up in the eyepiece, and then there's what's called an objective lens down at the bottom. You can see that in this modern microscope on the right. The ocular lens is up here where it says number 1, and then the silver things labeled number 3, those are different objective lenses. And the combination of those two lenses makes this for a much more powerful microscope. But even going back into the 1500s, people like Galileo was known to have had at least one microscope. Lots of scientists back then had them, and they got to look at very, very small things. The whole concept of the cell was advanced by a guy named Robert Hooke in the 1600s. And he was looking through a microscope at pieces of cork. Cork is actually the bark of a cork tree. And he noticed these little uniformly sized and shaped little structures, and he called them cells. And then turns out that all living things are made out of these cells. So I already mentioned one of the limitations of light microscopy, even though it just absolutely revolutionized science, from the Renaissance onward. There are some issues with it that kind of limit its usefulness. I already mentioned the fact that light can only reflect off of objects that are bigger than its wavelength. So that means that light microscopy can only resolve things or see things down to 10 to the minus 7 meters, or about 1/10 of a micron, which means you can easily see the structure of tissues. You can see how cells are arranged in tissues. You can see individual cells. You can just barely make out the organelles in the cells using light microscopy. So you can clearly see things like the nucleus, and even some smaller things like ribosomes. You get into some of the smaller organelles like ribosomes. I meant to say mitochondria before. You can see the nucleus, you can see mitochondria, but very small things like ribosomes might only appear as little tiny dots. You can't really see them very clearly. Things like bacteria are barely visible under light microscope. You can see a whole colony of bacteria, but the individual bacteria are very difficult to make out with light microscopy. And viruses are pretty much invisible to a light microscope. They're much too small. And certainly individual molecules can't be seen with the light microscope. Another downside to light microscopy, another limitation, is that in order to see anything, light has to be able to pass through the thing that you're looking at, which means you have to slice your specimen into very, very thin, microscopically thin slices. There's a thing called a microtome. It's a very sharp cutting blade. Oftentimes what scientists will do is they'll embed something in wax and then use this microtome to make extremely thin slices. And you put that slice on a slide and then look at it under the microscope. But the light has to literally be able to pass through this thin slice. Those slices are so thin that they're virtually transparent. And the structures you're looking at have no color. They're so thin that you can't actually see the color of the object that you're looking at. So oftentimes the only way to actually see anything with a light microscope is you have to stain it. You have to use stains that are attracted to usually to different molecules in the specimen that you're looking at. And then what you're seeing is actually the staining of those structures in these very, very thin, transparent slices. So I'm going to talk a little bit about staining techniques that are used with light microscopy, because again, as medical illustrators, these are things you're going to encounter all the time. And so I think it's very useful to have some understanding of what you're looking at when you look at a microscopic section of any kind of tissue or cells. By far, the most commonly used stain technique in the medical field is what's known as H&E staining. And H&E is short for hematoxylin and eosin. It's a combination of two different dyes or two different stains that are used to stain different things within the cells. So this stain is used widely in histology. I know the grad students are required to take histology in the fall. It's an option for undergrads if you want to take that. I highly recommend it because you'll learn all about the cellular structure of tissues and organs. And you'll just look at slide after slide after slide after slide of things that have been stained with H&E. If you do any kind of pathology as well, to look for cells that don't look right, cells that have some kind of disease, something wrong with them. Again, H&E is by far the most common type of stain that's used in pathology. So it's something you're very likely to encounter pretty frequently. So the H&E consists of these two different dyes or two different stains. The first one's called hematoxylin, and hematoxylin is a base. So it has a pH above 7. And because it's a base, it's attracted to acids. The base neutralizes the acid. So it's almost like positive and negative, are attracted to one another, like the different ends of a magnet. The acid and base are attracted to one another. So this basic or alkaline hematoxylin is attracted to anything that's acidic in the cell. And one place where you have a lot of acid in the cell is in the nucleus, because the nucleus is filled with deoxyribonucleic acid, DNA. DNA is acidic. As a matter of fact, when the nucleus was first discovered and DNA was first discovered, they had no idea what it was. They had no idea what function it had. All they knew was it was an acidic material in the nucleus. That's why DNA and RNA are called nucleic acids. They are just acidic stuff that's in the nucleus. So that was discovered using this type of stain. So when you stain a cell with hematoxylin, it stains the nucleus either very dark blue or very dark purple color. The hematoxylin itself has this purpley blue color. And so it's attracted to all of that acid in the nucleus. So whenever you see a microscopic section of slides and you see lots and lots of little purple dots, these are all nuclei and they've all been stained with hematoxylin. There's a type of white blood cell that you may or may not have heard of called a basophil. It's an important part of the immune system. And basophils have little granules in the cytoplasm that contain acidic material. And so this hematoxylin is also attracted to the cytoplasmic granules in basophils. In matter of fact, and so what it does is it turns these granules blue. So basophils are always recognized by having these little blue polka dots all throughout the cytoplasm. In fact, that's where basophils gets their name, because hematoxylin is a base. The word basophil means loves base. So basophils were first recognized and identified by the fact that they attracted this basic or alkaline stain. The other half of the H&E staining process is eosin. And I've never actually done this technique myself. I've never actually done the staining, but I know that you have to introduce one of them and then flush it out, and then introduce the second one and then flush it out. Because one of them is an acid and one's a base. If you put them both in at the same time, they would simply neutralize one another and do nothing. So you put in one, rinse it, you put in the other, and then rinse it again. So eosin, on the other hand, is acidic, which means, again, opposites attract. It's going to be attracted to anything that's a base inside the cell, but also it's particularly attracted to proteins. I don't know that proteins are actually a base, but for whatever reason, eosin is attracted to proteins. And it stains proteins bright red. So red blood cells, also known as erythrocytes, are called red blood cells for two reasons. Number one, because they're in blood, and blood is obviously red. But when you stain red blood cells with eosin, that eosin is attracted to all of that protein in the cytoplasm of the red blood cell. Because what's a red blood cell? It's a giant bag of hemoglobin, the protein that carries oxygen in your bloodstream. And so this eosin is attracted to this huge amount of protein within the cytoplasm of the red blood cell. So red blood cells stain red as well as looking red, when you just look at blood. Most other cells don't have as much protein in it. Not compared to a red blood. Like I said, red blood cell is just a giant bag of hemoglobin, big bag of protein. But other cells are going to have some protein kind of scattered throughout the cytoplasm. And so the red stain is going to look more pink because it's kind of diffuse. It's less intense. So most cells will, their cytoplasm is going to stain pink with the eosin. And then finally, there's another category of immune cells, white blood cells, called eosinophils, that are named for the fact that they attract eosin. Eosinophils have little granules in their cytoplasm, which are basic, they're alkaline. So they're going to attract the acidic eosin, and they're going to end up having red granules of cytoplasm in these eosinophils. So again, eosinophil literally gets its name. It means loves eosin. So they were recognized because of attracting the eosin stain to the cytoplasmic granules. So I got some pictures to show you so you can see what things look like with these different staining techniques. Here's what a typical H&E stained section looks like cut through a tissue. This happens to be skeletal muscle, so this could be, a slice through your biceps or your gluteus maximus or any other skeletal muscle. And if you look closely at the pink areas, you might notice that there's discrete sort of geometric shapes that are pink. All of that pink is the eosin that's lightly staining the proteins in the cytoplasm of all of those cells. These are actually the muscle cells themselves, the myocytes. They're cut in cross section though. We're not looking at myocytes in lengthwise. We're looking at them as though we've sliced across them in cut section. So every one of those little pink geometric shapes is the cut section through a muscle fiber or myocyte muscle cell. All the little pink dots are nuclei that are stained by the hematoxylin, stained dark blue or dark purple. So you see lots and lots of nuclei in here. In fact, one thing that's interesting about the skeletal muscle, if you would really get in there and count, you'd find that there are a lot more purple dots than there are pink geometric shapes. Anybody know why that is? Anybody know a unique feature of muscle cells? This might be something you talked about in an A&P class at one time, but I wouldn't blame you if you don't remember. Oh, Christine? Right, yeah, myocytes are multinucleate. Every single muscle cell has more than one nucleus. So there's actually, you would expect there to be more nuclei than you have cells. Here's another H&E stain. This happens to be lung. So some of these white areas are alveoli. Now they're not nice, round, perfect alveoli like we often think of them in the lung. I think a lot of that's just because they get distorted in the preparation process. But imagine, that this was a big round alveolus, filled with air. Surrounding the alveoli, there are some faint pink or sort of lavender colored cells. There are these very thin, flat cells lining all of those air spaces. These are what are called type 1 pneumocytes. They're the main cells that make up the walls of the alveoli. They're very flat, very thin, because they have to allow gas exchange. They have to allow oxygen and CO2 to pass through. Some of the purple dots represent the nuclei of those type 1 pneumocytes. And all of the red are red blood cells, called erythrocytes. Those are erythrocytes that are in the capillaries surrounding the alveoli. And they're the ones that are absorbing oxygen from the air and then dumping CO2 back into the alveoli to be exhaled. And over on the far left, we see a very large cluster of red blood cells. That's an arteriole. It's a little bit larger vessel, so bigger than a capillary. And here we see three types of white blood cells. that we talked about, basophils and eosinophils briefly. I'll go back over those in a second. Collectively, these three types of cells are all called granulocytes, which refers to the fact that they all have granules in the cytoplasm. The basophil is the kind that attracts base, and it's attracting the hematoxylin, which is the basic stain, and that's what's causing all these granules in the cytoplasm to turn very dark blue. The nucleus is in there too. It's a little hard to make out, but the nuclei of all three different types of granulocytes are all what we call multi-lobed nuclei. It's not just one circular nucleus. It actually has multiple lobes to it. And you can kind of see, I think there's a lobe here and a second lobe there and maybe a third lobe over there, all interconnected, forming almost like a little W shape in here. This one's even harder to make out, but we can see there's some purple in there for the nucleus. But the key thing here is those really dark blue cytoplasmic granules, which is all the hematoxylin. And here in the middle, we have an eosinophil. The nucleus is a little bit easier to make out here. We've got one lobe over there, a couple smaller lobes interconnected, and then another large lobe over here on the left. And then if you look at the cytoplasm, you see there's all these red dots in the cytoplasm. That's the granules that are attracting the eosin, giving it its name, eosinophil. And then there's a third type of granulocyte known as a neutrophil, because its cytoplasmic granules are pH neutral, hence the name neutrophil. They don't attract the base, and they don't attract the acid either. They don't attract either. So the granules, even though they're in there, they don't really stain. So you can't really see them very well. Yeah. Yeah, I mean, both of these things it's just a liquid. I don't know the chemical structure of it at all. All right, moving on. Another type of stain that some of you may have heard of. I don't want to sound sexist, but it's just a fact. Any women in the room should be familiar with this stain. The Papanicolaou stain, developed by Dr. Papanicolaou, more commonly known as the Pap stain. And this is the stain that's used in a Pap smear, which is what happens in a gynecological exam, where the OBGYN or nurse or whoever uses a little paddle to scrape some cells off of the surface of the cervix, smears them on a microscope slide, and then stains them with this Papanicolaou stain and looks at the cells. And specifically what they're looking for is to try to determine what kinds of epithelial cells are present within that Pap smear, because if there are certain kinds of epithelial cells, it could be indicative of like an early stage of cervical cancer. But the Papanicolaou stain is actually used more broadly. The Pap smear is the most famous application, and that's what it was originally developed for. But it can be used to recognize cells in any kind of smear of cells. Anytime you scrape some cells off, you can scrape them off the inside of your cheek, off the cervix, various places, spread them on a microscope slide, stain them with this stuff. But you can also stain cerebrospinal fluid. This is one way of detecting something like meningitis. Would be looking for the presence of white blood cells in the cerebrospinal fluid, which there shouldn't be. Sputum, so you cough up something from your lung, they can look at that, look for the presence of cells in mucus. Pleural fluid, they could, if you have fluid in your pleural cavity, they can aspirate some of that pleural fluid and look at it to see if there's immune cells in there. Even synovial fluid. They can analyze synovial fluid for the presence of things like neutrophils and macrophages, which would indicate some kind of immune issue going on inside of a joint. Because remember, synovial fluid is the fluid in a joint. So this might be, I'm not 100% sure about this, but I suspect this is probably a test for something like rheumatoid arthritis, where you have like an autoimmune disease affecting the joints. So the Pap stain consists of four different stains. So it's got your standard H&E. It's got hematoxylin, which is going to stain the nuclei blue or purple. You've got eosin, which is going to stain red blood cells red, but also any kind of superficial epithelial cells. Those cells that were like right at the surface of an epithelial membrane are typically going to stain either pink or red. Then you have a third stain called light green or fast green. This is going to stain any intermediate or deeper epithelial cells. And honestly, off the top of my head, I don't recall what the difference is between those kinds of epithelial cells, that one stains green and the other one stains pink. But that's what this stain is looking for. Some molecular marker that indicates, the presence of a deeper type of epithelial cell. And then there's a fourth stain called orange G6, and this stains keratin and glycogen. So keratin is protein that's found in skin cells, and glycogen is a long chain carbohydrate. Stains them orange. So here's one example of a bunch of cells stained with the Papanicolaou technique. So you got one up here, I'd kind of call that orange. So that could be almost like a skin cell, something from the dermis, which is staining kind of orange. We've got some other pink ones that would represent surface epithelium. And then we've got a bunch of these green ones, which represent the deeper layers of epithelial cells. Again, I don't know enough about the stain to actually look at this and tell you, is this person okay or not? I don't know. I don't even know exactly what is being stained. I don't know if this is a cervical smear or this is sputum or what it is. So I don't know exactly what it's showing, but it does show all of those different colors that would be revealed in a pap stain. Another one that's really very powerful stain, although it has a very specific use in the nervous system, is the silver stain. It uses silver to actually stain neurons. And there are two different versions of the silver stain technique. It was originally developed by a guy named Golgi, who you may have heard of, who is the Golgi complex or Golgi body is named after him. And he developed this technique of using a solution of silver to stain neurons. It was actually enhanced and developed further, perfected by a Spanish neuroscientist named Santiago Ramon y Cajal. Often just goes by the second of his last names is just Cajal, so it's referred to as a Cajal stain. So on the left, you see a typical neuron that's been stained with this silver stain technique, and you see it kind of looks red, but it's usually described as being kind of a reddish brown stain, or even in some cases even just solid black. And so the neurons just kind of pop out from all of the surrounding tissues. Because anything in the central nervous system is going to have lots of supporting cells. You have all these things called glial cells. So you've got astrocytes and microglia and oligodendrocytes. There are all these other kinds of cells within the central nervous system, but this stain is only staining the neurons themselves. So you can then see all the axons and the dendrites and the connections between cells. Yeah.
What is this? Is it just
That's a good question. There's something inside neurons called Nissl substance, NISSL, and I forget what the Nissl substance is made of, but I think the stain is binding to that. So it's something that's very specific to neurons. On the right is actually a drawing by Mr. Santiago Ramon y Cajal. In addition to being a pioneering neuroscientist, he was also a very talented artist, and he illustrated all of his own publications. And so what he would do is he would stare through the microscope at things that were stained using this silver stain, and he would draw these just ridiculously detailed, but also highly accurate pictures of the patterns of the different cells that he was looking at. I actually have a book with a collection of all of his artwork. That's it's really kind of interesting if you're a science nerd to look at this stuff. In fact, several years ago, I forget what year it was, the AMI held their annual meeting in Boston. And it just so happened that the MIT Museum in Cambridge was hosting an exhibit of his work. So I got to see all of his drawings, up close in person, and they're really pretty remarkable. So that thing on the right there is one single neuron. I believe that's called a MOXIE cell. I might be wrong about that. Another technique that's also used specifically in the nervous system, in fact, specifically in the central nervous system, is something called Luxol Fast Blue. It stains myelin. So myelin is a lipid that forms the insulation, around axons in the nervous system. And so this Luxol Fast Blue is attracted to that particular lipid, that fatty substance that makes up myelin. And so here we're looking at a cut section through the pons that's stained with a combination of H&E and Luxol Fast Blue. And so if you look at some of these cells, you see they have the characteristic H&E appearance. There's a dark purple dot in the middle, which is the nucleus, and then surrounded by some pink cytoplasm. And you see all of this pink in here is all cytoplasm of various kinds of cells. So these are all, some kind of supporting cells. These are astrocytes or oligodendrocytes or microglia, some of the other kinds of cells, but not specifically neurons. And then all of the blue stuff is the myelin. So those are all myelinated axons of cells in the central nervous system. And if you look in the upper right, you can see that we can actually see the sort of axons cut in lengthwise. So you see these long fibers of blue. Over on the left and towards the bottom, we see lots of little blue dots. What that's showing us is that we're actually looking at axons that have been cut in cross section. So we're just looking at the cut ends of all those axons. So they appear differently depending on whether you're looking at the cut ends or whether you're looking at them from the side, but it's all myelin that's stained with that Luxol Fast Blue. Right, another way of enhancing light microscopy. So everything we just talked about is using stains to make things more visible in a light microscope image. Another way to make things more visible in light microscopy is to use the principle of fluorescence. So when something fluoresces, what it means is that you are bombarding a sample with light, and that light has a particular wavelength to it. And the sample, whatever it is you're shining it on, the molecules are absorbing that light at a particular wavelength, but then they re-emit light at a different wavelength, always a longer wavelength. So they absorb light at one wavelength, and then they emit that light at a longer wavelength. So it literally causes the sample to glow. That's what glowing is. It's giving off a particular wavelength of light. Different wavelengths of light have different colors. In fact, that's what distinguishes red from green from blue. These are all just different wavelengths of visible light. One of the most common ways of getting something to fluoresce is to actually shine ultraviolet light. Now, ultraviolet light is a form of electromagnetic radiation that has very short wavelengths. The wavelengths are so short that you can't see it. Ultraviolet light is not visible to the human eye. Bees can see ultraviolet light, but humans can't. So you shine ultraviolet light on a sample, it absorbs that ultraviolet light, so it's absorbing energy, and then the sample then re-emits that light at a longer wavelength, which is something that does fall in the visible spectrum. So you shine UV on it, you can't see the UV, but you do see the green that's then emitted by the sample. So this is a common way of getting things to glow or fluoresce. There are three different categories of fluorescence, three broad categories. There's what's called autofluorescence, which I'll describe more in just a second. The second is called immunofluorescence, And then there's a third one, a couple slides coming up, called gene expression, or indicated markers of gene expression. We'll come back to that in a few minutes. So autofluorescence refers to the fact that there are several biological molecules that fluoresce all by themselves. If you shine the right wavelength of light on them, they will absorb it, and then they will re-emit a different wavelength of light. So you don't have to do anything special to them. They are autofluorescent, meaning they fluoresce entirely on their own. You just have to shine the right wavelength of light, and they'll fluoresce. So things like collagen and elastin, which are two proteins that are found in the extracellular matrix of a lot of tissues. They're very common in cartilage and in tendons, fascia, muscles, a lot of connective tissues. NADH is a molecule which is produced by the Krebs cycle, and it's a transporter molecule for electrons to what's called the electron transport chain. So it's a molecule that's present in the mitochondria. It's present everywhere, but it's present especially in the mitochondria, where it's used to help generate ATP. Tryptophan is actually one of the amino acids, so it's going to be found throughout proteins. And then chlorophyll, the photosynthetic molecule that's found in plants. These are all things that will fluoresce on their own if you shine the right wavelength of light on. The second type of fluorescence is what's called immunofluorescence. Let's say that there's some molecule that you're interested in inside of a cell. You want to be able to identify if the molecule is present, and you want to be able to see where this molecule is located within the cell. But it's not a molecule that autofluoresces. It won't fluoresce on its own. Somehow you want to create something that's going to allow that target molecule, that thing that you're looking for, somehow allow it to fluoresce. So what you do is you take a fluorescent dye, which is known as a fluorophore, and I'll talk a little bit more about them in a minute. There's a whole bunch of different fluorophores, or a bunch of different fluorescent dyes. You attach this fluorescent dye to an antibody, and you've actually manufactured antibodies that are going to target some particular molecule in the cells that you're looking for. So you might be able to create an antibody to actin, which is one of the fibers that that makes up the muscle, the sarcomere, muscle filaments. You could target a specific protein on the surface of a cell, some receptor protein on the surface of a cell. So you can manufacture an antibody that is attracted to pretty much anything you want inside the cell. So you attach this fluorophore, this fluorescent dye to it. You introduce that antibody. The antibody is going to bind to whatever that target molecule is. Then you shine some light on it. You watch it glow. You take a picture of it, and you then can see exactly where that molecule is present. That target molecule is present within the cell. The first of these fluorophores that was developed was, or really discovered, it's something that exists in nature, but scientists discovered it and started using it in this immunofluorescence technique. There's something called green fluorescent protein. It's actually derived from a fluorescent jellyfish, and you can now manufacture this protein and use it as a fluorescent dye. Now, that was back in the 90s that GFP was first utilized. Now it's very common. Any biology lab is going to have GFP on hand to do immunofluorescence. But now there's a whole bunch of fluorophores. So you can actually have different colored dyes attached to different antibodies and introduce these into a cell or into a tissue. And, one colored fluorophore its antibody is going to bind to one molecule. You could have a different fluorophore attached to a different antibody binding to some other molecule inside the cell. So you can get these really brightly colored images using all of these different kinds of fluorophores, such as these. So here are some images that are created by photographing cells or tissues after introducing these different fluorescent dyes. and bombarding them with some wavelength of light. So on the left, we have vascular endothelial cells. So these are the flat cells that line the inside of your arteries, veins, and your lymphatic vessels. And so you can kind of tell there's a big one here. It's very flat. It's like a thin, flat sheet. The blue in the center is some sort of dye, one of the, like the cerulean fluorophore, that is attached to an antibody that is specifically targeting something in the nucleus. Now, I don't know if it's targeting DNA or if it's targeting the histone proteins that the DNA is wrapped around, but there's definitely something in the nucleus that these antibodies are attracted to, and they're glowing blue because of the blue fluorophore. All of these kind of greenish strands, those are strands of actin. Actin is one of the proteins in the contractile unit of the muscle cell, but actin is also really important in all cells because it's one of the main components of the cytoskeleton. These are structural fibers that help to give the cell its distinctive shape. So all of those green strands are actin. And then there's some kind of red fluorophore that's targeting something probably on the surface of the cell, something on the cytoplasm on the outside. So we've got three different antibodies targeting three different molecules in the cell, each one carrying a different colored fluorophore. So you get these vivid, brightly colored pictures. On the right, we're looking at a cut section through smooth muscle. The round things that you can see here, round or oval things, those are actually blood vessels in the smooth muscle. So blood vessels that are supplying blood to this muscle. And all the green is attracted to the actin filaments within those smooth muscle cells surrounding these arteries. And I'm not sure what the red is that surrounds that on the outside, or what this kind of, I don't know what you call that, peach color inside. I mean, that's where the red blood cells would be in there. So something must be targeting those red blood cells inside the lumen of each of these vessels. Here's another one that I think is kind of cool. Actually captured a cell in the process of mitosis. You're going through cell division. So we've got one antibody with a blue fluorophore that's attracted to the chromatin in the chromosomes. So these are the chromosomes lined up on the mitotic spindle. You've got another antibody and it's kind of a gold-colored fluorophore that's attracted to the mitotic spindle. The mitotic spindle is made up of a protein called tubulin. So this is somehow attracted to that protein. And then there's an antibody that's attracted to something in the cytoplasm, giving us all of this green stuff around the outside of the cell. And so you can see that this cell was captured right in just about the completion of cell division. And here's one I just thought was really pretty. I'd like to put a print of this on my wall. This is gastric mucosa. So showing that different kinds of cells within the mucous lining of the stomach express all different kinds of molecules. And so there are antibodies then to these different molecules, each one carrying its own different fluorophore, its own fluorescent dye. So we've got something red up here that's staining the gastric pits. Then we've got, some bright yellows and greens staining other things deeper in the mucosa, and then down here into the submucosa. So just, really cool image. It really reveals how you can target different cell types. All right, and the last type of fluorescence, the last use for fluorescence in light microscopy is what are called reporters of gene expression. So this is not designed to target some molecule in the cell. This is designed to detect when a particular cell is expressing a certain gene. That's something that's often of interest to biologists. They want to know which cells express gene X, or at what point in their development does this group of cells express gene X? And then at what point do they maybe stop producing gene X? So what you do is you genetically engineer some cells. You take the gene that you're interested in, and you actually insert the gene for green fluorescent protein into that gene that you're looking for, that you're interested in. So this little graphic here shows we've got the gene for green fluorescent protein, and it's a very small protein, so the gene's not very big. You actually insert that into the DNA of some other target gene, some gene that you're interested in. And so then when the cell transcribes and translates that gene into protein, you get the main protein that the gene is supposed to produce, but it actually has green fluorescent protein attached to it. And that green fluorescent protein will then fluoresce. You shine ultraviolet light on it, and it's going to glow green. So you can use this then to, like I said, determine which cells in a particular sample are expressing whatever this gene is that you're interested in, and also timing. You know, at what point in development does it express this gene, and does it continue to express this gene throughout its life, or does it stop expressing it at some point in development? So over here on the right, we have a photograph. This is just a regular light microscopy photograph, but with the addition of fluorescence. This is the cerebral cortex of a mouse, so part of the brain of a mouse. And we're looking to see which cells in the mouse cortex are expressing a gene called MAPT, which is the tau protein, which is an important protein that's implicated in Alzheimer's. One of the features of Alzheimer's is the accumulation of excess tau protein. So if you're researching tau and you're researching Alzheimer's, then one of the first things you want to know is which cells express tau in the first place, and at what point in their development do they express tau, and do they ever stop expressing tau at some point? And what happens if you have a mouse with Alzheimer's? Does it express more of the tau, or do certain cells express it that normally wouldn't express it? You know, so I don't know exactly what they were studying in this particular mouse cerebral cortex, but they're looking specifically at the expression of this tau protein. All right, so why don't you take a little break, because I've been talking for a long time. Take 10 minutes or so, and then we'll resume. All right, the next type of microscopy that I want to talk about is electron microscopy. This is a much more recent development, first pioneered in the 1930s and getting progressively better ever since. And it takes advantage of the fact that electrons have a much shorter wavelength than visible light. And again, I mentioned this before, we don't normally think of electrons as having a wavelength. We think of them as a type of particle, but they do actually have a type of wavelength. The wavelength of the movement of an electron is about 10 to the minus 9 meters. So that means it's about 100 times smaller than the wavelength of visible light. So that means that electrons can be used to resolve things down to about 10 to the minus 8 meters, or roughly about 10 nanometers in size, which means that you can now see not only whole cells, but you can clearly see the individual organelles. You can clearly see bacteria. You can even see viruses. There are some downsides to electron microscopy. It's a very complex process and requires some very difficult preparation to get samples ready for electron microscopy. For one thing, the sample has to be completely dehydrated. There can't be any water present because the water will disrupt the electric current that's actually used for creating the image. But you have to somehow dehydrate the objects without them losing their shape. I mean, you think about most things that get dehydrated, they shrivel up and they're unrecognizable. So what you have to do is you actually have to replace the water with a different solvent that does not conduct electricity. So something like replacing all the water in a specimen with something like acetone or some other kind of liquid that's going to allow the thing to keep its shape, but without being electrically conductive. The second thing that you need to be able to do, or that you need to do when you're preparing something, is after you've dehydrated it, got rid of the water, replaced it with some other liquid, you have to coat the surface of the sample with a very thin layer of electrically conductive material, which is usually gold. When I first started teaching this class, I used to bring students right upstairs to the second floor of this building. There's actually an electron microscopy lab, or used to be. They had a couple of old electron microscopes that I think have finally died, and they don't have the hundreds of thousands of dollars that they need to replace them. So I don't think that it exists anymore. But Ted Kinsman, faculty in the photo school, used to run this lab, and he would demonstrate the process of what's called sputter coating, where they would take a tiny, tiny little piece of gold and put it into this machine that would vaporize the gold, turn it into a very fine vapor of gold metal particles, which would then settle onto the surface of the specimen that they were trying to image, creating this thin coat, almost like a single atom thick of gold on the surface of the specimen. He would always, just for kicks, he would take like a penny from somebody and he would sputter coat it with gold so it looked like he had a gold plated penny. Of course, the amount of gold on that was probably like less than a cent worth. The penny itself was worth more, but that was a very complicated process. The other downside, which is not that big a deal, but it's just something to be aware of, is that all electron microscopy images are grayscale. There is no color, because the color that we see in the world is all coming from visible light. And remember, the whole point of electron microscopy is that we're using something that has a wavelength that's much shorter than visible light. So the whole concept of color is irrelevant at this scale. There are three broad categories of electron microscopy that you should be familiar with. The first is what's called scanning electron microscopy. And this looks at the three-dimensional surface of an object. So this is the one where it's really particularly critical that you remove the water while retaining the shape, and you do that sputter coating with a layer of gold atoms, because what you're trying to do is preserve the three-dimensional shape of some kind of surface texture. And you end up producing these incredible images that appear to be like a three-dimensional landscape inside the body. These things are typically the lowest resolution. The resolution of SEMs is usually comparable to light microscopy. SEM doesn't allow you to get the really fine detail that some of the other techniques do. The next technique is called transmission electron microscopy. And this is similar to light microscopy in the sense that you have to create a very, very thin slice of a material, very thin slice of a cellular tissue, whatever, and you're imaging a cross-sectional view of it. Now, this particular type of microscopy has much higher resolution. So this is now getting about 10 times better resolution. Now you're getting down to that 10 to the minus 8 meter scale. So down to like, 10 nanometers in scale. And then the final type of electron microscopy, which is a much more recent development, just literally within the past couple decades, is what's called cryo-EM or cryo-ET. Cryo-EM is simply cryo-electron microscopy. Cryo-ET is short for cryo-electron tomography, which is a three-dimensional form of electron microscopy. The prefix cryo means cold. And the way that this works is that you have to get the sample down to extremely low temperatures. And at very low temperatures, objects become very conductive to electricity. And so that allows you to do electron microscopy that has incredibly fine resolution on these things that are chilled down almost to the point of absolute zero. So this allows you to get the finest resolution all the way down to about 10 to the minus 9 meters, so down to nanometer scale. So now we're almost down to the scale of individual molecules. So here's a scanning electron microscopy image. And as I mentioned before, these don't get super high resolution. They're about the same as a light microscope, but they allow you to create these three-dimensional surface images. You can actually image the 3D surface of a tissue or an organ or an individual cell. So here we're looking at the inside of a bronchus, and you can see lots of villi or cilia. These would be the little cilia that would sweep mucus up out of the bronchus. There are various kinds of cells. I know these kind of star-shaped cells, those are macrophages. I forget what the other cells, the more rounded ones, are in here, but you can see, various kinds of cell types. Of course, you see that it's all grayscale because, again, all electron microscopy images are going to be grayscale. Here's one that's very high resolution. It's still scanning electron microscopy, but we're actually looking at the surface of an individual bacterium, an individual E. coli cell. That's what this gray thing is back there. And these little lighter gray things, those are bacteriophages. Those are individual viruses that are infecting this E. coli, injecting their genetic material into this E. coli cell. Yeah. Yes. Yeah, electron microscopy is the only way you're actually going to see viruses. Here's kind of a fun one. This is another scanning electron microscope image. So we're looking at a three-dimensional surface of a structure. We're actually looking at the underside of a cannabis leaf. And it's got these interesting little spiky things that look like thorns, but the important parts are these things called trichomes, these little stalks that extend down from the underside of the cannabis leaf. And the little purple blobs are little droplets of THC, which is the active ingredient in marijuana. The plant actually exudes these little droplets of THC as an insecticide. THC is meant to kill bugs or insects so they don't eat the plant. Now notice the little note down here in the bottom right. This is false color. This is the same guy who ran the electron microscopy lab upstairs. This is Ted Kinsman's work. And he hand colored this in Photoshop. So the original image is purely grayscale. And then he went in and just, selected areas and colored them in Photoshop. Yeah. Oh, question. Yeah. This is still scanning electron microscopy because we're looking at a three-dimensional surface view of something. And this is relatively low resolution, in the grand scheme of microscopy. This is actually Ted's book, in case anybody's looking for an interesting Christmas present. Anybody who's a real pothead and wants some pretty pictures of cannabis under the microscope. Now, here's an example of transmission electron microscopy. So this is something that's actually utilizing thin slices through something. This is a thin slice through a plasma cell, which is a type of cell involved in the immune system. And so this is incredibly high resolution because we're looking at the nucleus here. This giant kind of roundish thing is the nucleus. Everything labeled M, those are mitochondria. So you can see the mitochondria pretty clearly. And all of this squiggly stuff out in the cytoplasm, that's all endoplasmic reticulum. And if you look closely, you may be able to see that all of the endoplasmic reticulum has little dots on it. So that's all rough endoplasmic reticulum with ribosomes on it. So we're looking here at what's called a plasma cell. B cells or a certain group of B cells will convert over to plasma cells, and then a plasma cell is basically just a little factory that produces antibodies in response to some infection. And plasma cells have this very distinctive appearance to them. First of all, the nucleus is always off to one side. So notice how the nucleus is not right in the dead center of the cell. I mean, you can't see the whole cell, but I think you can still tell that the nucleus is shifted slightly to the left in this image. And then there's more cytoplasm on the right. It's referred to as an eccentric nucleus because it's not centered. The nucleus also has this very weird pattern. All that black stuff is chromatin. The chromatin has condensed into these clumps. And there's almost always one big central clump right in the middle, and then a series of clumps around the outside, kind of radiating outward with these lighter colored patches in between. somebody thought that this looked like a wheel with a bunch of spokes. So this is actually known as the wagon wheel nucleus. So these are two distinctive features of B cells. And I've illustrated lots and lots of these. So I learned very quickly how to create a very stylized B cell that people would still recognize immediately. You put the nucleus off to one side, make basically like an egg-shaped cell, the nucleus off to one side, put the little wagon wheel spokes, the nucleus, and people will, at least, immunologists will immediately recognize it as a plasma cell. Right. The final type of imaging that I want to just mention very briefly is something called X-ray diffraction. This was actually discovered, interestingly enough, it was discovered before electron microscopy. So this goes back to the 19-teens. And it takes advantage of the fact that X-rays have even smaller wavelengths than electrons. X-rays have a wavelength down around 10 to the minus 10, 10 to the minus 11 meters. So very, very small. So that means they can reflect off of just about anything. And you can use X-ray diffraction to image molecules or even individual atoms, things that are on the scale of 1 angstrom, which is 10 to the minus 10 meters. So what you're looking at on the bottom left is what's called an electron density map. It actually shows where there are clusters of electrons, which are going to indicate the presence of individual atoms, because an atom is basically just, a bunch of electrons whizzing around a nucleus. So this sort of topographic map shows us where all the electrons are located, so we can tell that there are atoms located at each of those little peaks on the topographic map, and that's actually an electron density map for this particular molecule over on the right, which I forget what that is off the top of my head. But we're going to come back and spend a lot more time talking about X-ray diffraction later in the semester when we get to the molecular visualization portion of the course, because that's by far the most common tool that's used for determining the 3D structure of molecules. For much of the 20th century, anyway, there was something known as a resolution gap. Remember that light microscopy is very ancient. It goes back to Renaissance times, back to the 1500s. But even with further refinements in light microscopy, it hit that theoretical lower limit. Light microscope cannot theoretically see anything smaller than 10 to the minus 7 meters, down to about 1/10 of a micron. Which meant that you could see cells, you could kind of see organelles, but you couldn't really tell anything about the organelles. You couldn't really determine their detailed structure. And then in the 19-teens, along comes this thing called X-ray diffraction, which allows us to see. 1 second. Oh, sorry. Another question. Yeah. So X-ray diffraction is used for imaging molecules, and it works by actually crystallizing the molecule. You actually have to have a pure crystal of the molecule that you're trying to study with X-ray diffraction. So you're not actually shining the X-rays into any kind of living tissue. So you're absolutely right. If you shine these X-rays into a living cell, you would probably kill the cell or cause it to mutate in some horrible way. But they don't use the X-ray diffraction on actual living tissues. You actually have to isolate whatever molecule you're interested in, get it to crystallize first, and then you do the X-ray crystallography on that, or X-ray diffraction on that. All right, so like I said, X-ray diffraction was developed in the teens and really took off throughout like the 1930s, the 1950s. The DNA, the structure of DNA was determined in 1953. Protein structures in the 1960s. And X-ray diffraction allows you to see things that are very, very small, things that are smaller than a nanometer, smaller than 10 to the minus 9 meters. So it allows you to determine the three-dimensional structure of individual molecules. But for much of the 20th century, there was this gap in between. There were light microscopy could go down to about 10 to the minus 7 meters. X-ray diffraction allowed you to see things that were 10 to the minus 9 or smaller, but nothing in between. There was a whole range of sizes in that 10 to the minus 7 down to 10 to the minus 9 meter scale that we simply couldn't see, which meant you could just barely see organelles, but you couldn't determine the 3D structure of the organelle. You could see molecules, individual molecules, but you couldn't see how the molecules were packaged together to form bigger structures in the cell. So that's kind of represented by this image here, where again, we see the scale running across the top. We got a meter, a decimeter, which almost nobody uses that term, centimeter, millimeter, 100 microns, 10 microns, 1 micron, 100 nanometers, 10 nanometers, 1 nanometer, 1/10 of a nanometer, which would be the same thing as an angstrom down here at 10 to the minus 10 meters. So we've got, height of a kid, thickness of a human hair, size of a red blood cell, etc., all the way down to the size of individual atoms and molecules down here at a very small size. So the human eye can see down to 10 to the minus 4 meters. Light microscope can extend that down to 10 to the minus 7 meters, which is the theoretical lower limit of light microscopy. X-ray diffraction can see things from 10 to the minus 9 or smaller, but for much of the 20th century, you had this resolution gap where we couldn't see things in here. And then along comes electron microscopy, and especially cryo-EM and cryo-ET, which finally, literally just within the past couple of decades, has filled in that gap, that resolution gap. So now we can actually see the detailed structures of a virus. We can see individual molecules, but importantly, we can determine how molecules are actually packaged together to form organelles within the cell. So now we're able to see the full range of different scales. So this cryo-EM, cryo-ET, these are things that allow us to bridge that resolution gap. Like I said before, it's simply performing electron microscopy at very cold temperatures, which increases the electrical conductivity of the sample. The difference is that cryo-EM is going to create just individual cross-sectional slices. With cryo-ET, you take those cross-sectional slices, you stack them up to create a 3D model. It's exactly the same as taking a bunch of cross-sectional images from a CT scan and stacking them up to create a 3D volume model of some anatomic structure, just like we did with Horos. So cryo-ET is going to give you these 3D reconstructions based on a series of cross-sectional electron microscopy images. So here are some of the things that have come out relatively recently utilizing cryo-electron tomography. On the left is a picture of a ribosome. And you can actually see how various molecules are packaged together to create the structure of the ribosome. So a ribosome always consists of two subunits. There's a small subunit shown in yellow. This is where most of the business of the ribosome happens. And then there's a larger subunit shown in light blue. These two subunits are stuck together. The messenger RNA, which is transcribed in the nucleus, messenger RNA comes out into the cytoplasm, finds a ribosome, and enters the ribosome up here. You can actually see this is what's called the 3' messenger RNA entrance, where the messenger RNA is coming into the ribosome. You can see a little chunk of messenger RNA, and then it comes back out of the ribosome over here on the left. So we're actually seeing a little chunk of messenger RNA as it passes through the ribosome. And then inside the small subunit of the ribosome, these three colored things, there's kind of an orangey brown one, a green one, and a purple one. Those are all molecules of transfer RNA, tRNA. Do you remember in the process of translation, there's a 3-letter code on the messenger RNA called the codon. And then the tRNA has a matching code called the anticodon. And the tRNAs come in and the anticodon binds to the codon. And so you have one tRNA molecule that's about to get kicked out of the ribosome. You have another one, the green one, which is in, I forget, the sites have names. It's like the P site, E site, A site, I think. The green one is actually bound. Its anticodon is bound to the codon of the mRNA. and delivering its amino acid. And then the purple one is at the entrance. That's the one that's about to enter to deliver its own amino acid. And then just barely visible is a little brown thing down here, extending down from the 3 tRNAs. That's actually the protein, the little strand of protein that's being manufactured from the amino acids that are all being stuck together. That protein is actually going to pass down through this dark blue thing called a translocating PCC. And shoot, I keep forgetting what that's called. Protein conducting channel, I believe is what it's called. It forms a little pore in the surface of the endoplasmic reticulum. And the protein that's produced by the ribosome then passes through that little pore, that protein conducting channel, down into the endoplasmic reticulum, where it's then stored until your body needs, whatever that protein is. Oh, okay, I just realized I put it in here so I wouldn't forget what it was. That's what PCC is. Right, so that's a really detailed view of the detailed structure of a ribosome, something that we could never see before the development of cryo-ET. On the right is the structure of a microtubule. A microtubule is also part of the cytoskeleton of the cell. There are microtubules that just help to hold the shape of the cell, but microtubules are also important because they form the mitotic spindle. And microtubules are actually made up of two proteins called alpha and beta tubulin that simply spiral around. You just have one alpha, one beta, one alpha, or one beta, and they just kind of create this helical spiral and create these long tubes of protein. This is something called a perforin, which is something that can actually punch holes in the surface of a cell. At the top, you actually see some of the cryo-EM images, some of the individual cross-sectional slices, and then those things are stacked up to create this 3D model down here. So the yellow color actually represents a cell membrane of some kind of organism, and then the blue thing is this protein complex that punches a hole in the surface of that cell membrane. On the right is are the contractile proteins in muscle and the sarcomere of the muscle. The red in the middle is actin, which is what we call the thin filament in the contractile unit of muscle cells. The blue ribbon that kind of spirals around the actin, that is called tropomyosin. It's another protein. When a muscle gets ready to contract, what happens is that calcium ions come in and bind, causing a shape change in the tropomyosin. Tropomyosin moves to reveal binding sites on the surface of the actin molecule, and that allows myosin heads to then attach to the actin. And actually, all these other colors, the kind of pale yellow, the blue, the purple, those are all parts of the myosin heads that are now binding to the actin molecule. So you're actually seeing how myosin binds to actin to create contraction of muscles. And here's an even more recent publication that shows this. This just came out last year, I believe. Somebody did some cryo-ET on the entire sarcomere of a muscle, the thick filaments, which are the myosin, and the thin filaments, which are the actin. And so, and they even show it organized by, like, there's a P zone and M band and all these different parts of the sarcomere. And you see the individual components, the individual molecules over here. I think this next picture is even more dramatic. It's kind of a 3D view of the same thing. So the bright yellowish green, again, that's the actin molecules that form long strands. So there's one there and another one up here. And then the light pink ropes that are wrapping around the actin, that's the tropomyosin. And so the calcium ions bind to that and cause it to move to expose binding sites for the myosin. That all this blue and purple up here at the very top, right? That's all the myosin thick filament, and then these yellow things, these are the heads, the myosin heads that are actually binding to the actin filaments, and that's the part that's actually going to contract to create muscle contraction. Right, so summarizing everything that I just talked about, we've got light microscopy, which allows us to see things down to the scale of about 10 to the minus 7 meters, which lets you easily see the structure of tissues and easily see individual cells. You can dramatically enhance light microscopy using vital staining methods like H&E or Luxol Fast Blue or some of the other ones I talked about. You can also enhance light microscopy using the principle of fluorescence, either autofluorescence, immunofluorescence, or reporters of gene expression. The electron microscope extends our ability to see things down, another order of magnitude, about 10 times smaller, so that we can now see things. We can see cells very clearly. We can even see individual organelles. You can see bacteria and viruses. Cryo-EM and cryo-ET extend that resolution even further, down to 10 to the minus 9 meter, down to nanometer scale. So you can see the detailed structure of things like organelles and viruses. You can actually see how the individual molecules are packaged together to create different organelles or even to create an entire virus. And then finally, X-ray crystallography. That should actually say 10 to the minus 10, not 1 to the minus 10. This allows us to actually determine the three-dimensional structure of individual molecules. All right. To actually illustrate cells, there's some different references that I've included here, and I put direct links to these in my courses if you're interested in going and looking at them. I'm not going to bore you with going to look at them. We will come back and use the electron microscopy data resource. I think next week we're actually going to do an exercise in class where we use that as a resource. But maybe I'll demonstrate some of these things for you next week before we do that exercise in class. But they're handy references to have if you want information about cells or cell structures. The BioNumbers database is interesting. It's not pictures, it's all numbers. Like if you want to know what's the diameter of a typical red blood cell, you go there and you search for erythrocyte and it'll tell you the standard diameter, the volume, even things like the lifespan, like how long does a red blood cell actually live in circulation, stuff like that. So there's all kinds of interesting numbers in that. And then the rest of them are more image databases. But the thing I want to finish with today is just giving you some pointers about illustrating cells. And we'll use these for at least one exercise that we'll be doing a little bit later in the semester. A very common question that every medical illustrator has to ask themselves if they're illustrating something at the molecular or at the cellular level is what color to make the cells, because you don't really know. You don't have much to go on. Remember that light microscope images have virtually no color. They're basically transparent, thin cross sections. The only way you can see anything on a light microscope image typically is with either a stain or with fluorescence, in which case you're seeing the color of the stain or you're seeing the color of the fluorescent fluorophore, but you're not necessarily seeing the color of the cells themselves. Electron microscope images are all grayscale. Scanning electron micrographs, transmission electron micrographs, cryo-EM, cryo-ET, these are all grayscale. Now you can colorize them if you want, like Ted Kinsman did with his cannabis pictures, but the images themselves are all grayscale, so you don't really have any guidance about what to use for color. So there are some common approaches. I'm sure there are others beyond what I've listed here. Really, you're kind of wide open. You can color the cells just about anything you want, but I thought I would just share with you some examples of some approaches that I see used commonly and approaches that I've used myself. So the first approach is pretty straightforward and simple. If you got to illustrate cells, illustrate the cells to match the color of the tissue that they're part of. Very common approach. And again, I've done this myself many times. A great example of that is liver cells. If you look at a microscopic section of hepatocytes or liver cells, again, they're going to be completely clear unless you stain them. And they're usually going to be stained with H&E, so they're going to be purple and pink. But, we'll come back to that in a minute. You could color them that way, but you could also just color them to match the color of a liver, which would be dark brown or sort of dark purpley brown color. So here you see some pictures of the cellular structure of the liver where all of the hepatocytes are all colored brown to match the color of the liver itself. I think this is a terrible image, frankly. These are supposed to be myocytes, and it's somebody who clearly wasn't a trained medical illustrator who created this, but and I just swiped this off of Getty Images. It's just off a stock site, but pretty typical thing. If you're coloring muscle cells, then maybe you make them red because that's the color of muscles. Here's one from my own work. This is actually showing the pathology of celiac disease. And so what we're looking at here are enterocytes, or the epithelial cells that line the intestines. So I made them intestine color, kind of a peachy pink color to match the intestines. Here's from somebody else's work. I don't know who the artist is here, but on the right, we see gallbladder and bile ducts, the whole biliary system, in a kind of a pale green, yellowy green color. So in this enlarged view, we see the liver cells, the hepatocytes, and actually that kind of grayish color of the hepatocytes matches the color of the liver on the right. And then the bile ducts, these are bile canaliculi, and then a bile duct over here, using the same green color as the gallbladder, the bile ducts on the right, which makes it very easy to, kind of match up the colors you're seeing on the left with the colors on the right. So the second approach that you can try, and I know lots of people who use this technique, is to color cells to match the staining method that was used to visualize them. Now, this is a classic example here where we're actually showing the cell types that are distinguished by H&E staining. The basophil, the eosinophil, the neutrophil, the three different types of granulocytes that are distinguished by their attraction to either eosin or hematoxylin. So this is one where it kind of makes sense to match the staining method. And, so we've got our eosinophil with the bright red eosinophil stained granules, and the basophil with the dark hematoxylin stained granules. Neutrophil, I'd kind of like it if you could actually tell that there were granules in there, because there are granules. They're kind of clear, so they don't show up very well in this image. And then things like monocytes and lymphocytes, they don't have any granules in their cytoplasm. They're distinguished mostly by the size and shape of their nucleus. And then you got some erythrocytes or red blood cells in the middle. But this is exactly how all of these cells would look if they were stained using the classic H&E staining technique. And here's my own version of exactly the same thing. So this is an illustration I did for a medical terminology book, where I simply matched the stain. So we've got the classic erythrocytes up here. It's a biconcave disc. And we've got our eosinophil with red granules, basophil with blue granules, and a neutrophil with really kind of neutral granules, and then lymphocytes and monocytes. Lots of people, have done pretty much the same image. This one you can tell is done in 3D. These were done in Illustrator. There's another approach that you see pretty commonly, which I like to call the water balloon approach, where you illustrate cells as being basically just big bags of water, which is really not a very accurate representation, because there's a lot of stuff in a cell. There's a lot of molecules in the cytoplasm. The cytoplasm is just chock full of proteins and other kinds of molecules. So this idea that a cell is just this big bag of water with a nucleus in the middle is, I don't know, a little bit misleading. But you do still see people who who use this kind of approach. Oftentimes the cells appear blue because we associate the color blue with water. You know, the ocean looks blue, and sometimes you look at ice and it has like a bluish tint to it. And so we associate the color blue with water. So you'll often see clear bags of blue water to represent cells. And I've been guilty of that myself too. This is an illustration that I did for a book that I illustrated six editions of this book, I think, over a span of almost 30 years. They come out about every 5 years. So illustrated lots of editions of this book. It's the full name of it is actually Robbins' Pathologic Basis of Disease. Stanley Robbins was a pathologist at Harvard at Brigham and Women's Hospital back in like, I don't know, the 50s or 60s or something. And he wrote this book, and there were 5 editions of it before I got involved in it. And all of the artwork was like all just black and white. And then the publisher hired me to completely redo all of the artwork for the 6th edition, creating all full color, all done in Illustrator. And so it's a pathology book. So it deals with lots and lots with the immune system and immune cells and things. And so I had to think, all right, how do I represent all these different kinds of immune cells? You got B cells, and T cells, and plasma cells, and natural killer cells, and monocytes, and macrophages, and neutrophils, and eosinophils, and basophils, and all these things. So I went with just kind of your basic water balloon kind of approach, but the different kinds of cells are distinguished from one another primarily because of things like the nucleus, like this thing on the far left that has a bacterium inside of it. That's a macrophage. You can tell by the kidney bean shaped nucleus. This thing right here that has a 3-lobed nucleus, that's the classic look of a neutrophil. T cells and B cells are small round cells with a very large nucleus, where the nucleus occupies almost the entire interior of the cell. So, you know, little round guys with a very large nucleus. So, you know, everything had this kind of water balloon approach. And then, you know, like I say, distinguishing the cells just by size, shape, and the shape of the nucleus and some other key things. So I did that for the entire 6th edition, did hundreds and hundreds of illustrations for the 6th edition, came back, did a whole bunch more illustrations for the 7th edition, all in this same kind of style. And then I had a meeting with the authors. And so that's me, obviously. These are the 3 authors over here. That's Dr. Kumar, who was head of pathology at the University of Chicago. Dr. Abbas, who was head of pathology at the University of California, San Francisco. That's Dr. Astor in the background. He's at Harvard and Brigham and Women's Hospital. And then these 4 people are all representatives from Elsevier, from the publisher. And we had a meeting. They flew me to Chicago, put me up in a fancy hotel for a couple of days. If you ever want to know why textbooks are so expensive, let me tell you a story. And I put this in here just for fun. But so we got how many people here in this picture? 8 people. So we're all in Chicago at a fancy hotel. We actually worked. I mean, we spent like the entire day. They flew me in on a Friday. I spent Friday and Saturday nights. We spent like all day Saturday just in a room, in a conference room in the hotel, just talking about different things about this book. And then they took us out for dinner that night to a place called Charlie Trotter's, which I think doesn't exist anymore, but it was like a Michelin rated 5-star restaurant in Chicago. It's like the most expensive restaurant in the entire city. And it's one of these places where they don't have a regular menu. You don't order off of it. They have a menu. You go in there, you get everything on the list. So we had tempura of Nantucket-based scallops with togarashi and fuyu persimmon. I don't even know what half those words mean. Santa Barbara sea urchin with Buddha's hand, ginger, and horseradish. I mean, every one of these things. Of course, each one was like 1 spoonful, right? And they decided they would get the wine menu too. So we got a different wine with every course. And for 8 people, I think the bill was about $3,000 for dinner.
So 3 books.
So
Make a meal.
this is the most popular pathology textbook in the world. It's translated into 13 languages. So that's why they pull out all the stops for it. But still, it's actually it's the book is still surprisingly cheap. I think it's just a little over $100. So 30 books for dinner. But anyway, the real point of this, besides just telling you an amusing anecdote, is that at this meeting, not at the dinner, but at the meeting earlier, Dr. Kumar looks at me and he says, "Jim, I'll never forget this. Jim, why blue?" And I proceed to explain the whole sort of water balloon metaphor and how we associate the color blue with water. And I thought that seemed like as good a choice as any. And he says, I think I would have picked any other color besides blue. Like, well, why didn't you tell me this 2 editions ago? Why are we waiting for this meeting now? So we brainstormed and decided amongst ourselves that I would redo all of the art in the entire book to match H&E stain. So a light pink cytoplasm, which would be typical in H&E, and then dark sort of purple nuclei. Still distinguished by the presence of granules and other features in the shape of the nucleus and stuff like that. But now with pink cytoplasm instead of the blue. And this worked for 2 more editions. So I redid all the art for the 8th and the 9th editions. And then we had another meeting, although unfortunately this one was not in Chicago at a 5-star restaurant. This was just like a Zoom meeting. I think this was before Zoom. But anyway, we had another meeting and they said, we've gotten some feedback from the students that they still can't tell the different cell types apart. Which I get, because they're all the same color. So they decided that we would just go with a completely different approach where we would actually color code the cells so that all the B cells are one color, all the T cells are a different color, all the dendritic cells and phagocytes and whatever, they're all different colors. And which I don't know, aesthetically to me it reminded me of like a children's birthday party with lots of colored balloons. I wasn't necessarily thrilled with it from a purely aesthetic point of view, but I think from a didactic point of view, educational, like if the students have an easier time distinguishing the cell types because they're all different colors, then if that helps them learn, then it's probably a good thing. So we did that for the 10th edition. No word on the 11th edition yet. They want to completely change that again. Another example, this is the same book, just different picture. So this is the original water balloon approach from the 6th and 7th editions. The H&E stain approach from the 8th and 9th edition. Now, one thing actually another thing we changed in the 10th edition is they decided they wanted a few that were, I don't know, a little fancier, a little sexier. So instead of just the flat 2-dimensional Illustrator style stuff, I brought them into Photoshop just to, put some shadows and highlights and give them a little more 3D pop. So we did, I don't know, 20 or 30 illustrations like this where we gave them this little bit more 3D kind of appearance, kind of scattered throughout the book, like one in each chapter or something like that. So last thing I'll share with you is just a kind of a radically different approach to color. Way back on day 1, I showed you a little book by this guy, David Goodsell. He's a molecular biologist from Scripps Research Institute in California and does these remarkable watercolor paintings of the inside of cells. And in his case, I think his whole rationale is just there are so many molecules inside of these cells. You need some sort of color coding system just to tell them apart, just to distinguish them. I don't know that there's any other real sort of thought process behind the colors. Like on the left-hand side, this yellow strand, this yellow thing looks like a rope. That's actually DNA. why is that yellow? Let's see if I can identify something else in here. I'm having a hard time telling what's what. Let me look at this other one. I know more of what's going on here. The green little strands, these are all cell membranes. That's the outer cell membrane of this particular cell. This is mitochondrial cell membrane in here. So all the green are cell membranes, and then there's lots of little green blobs embedded in the cell membranes. Those are all proteins. So various kinds of receptors and channels and things embedded in the membranes. Again, the yellow rope, that's DNA again. There's some actin, this blue strand up here. That's a strand of actin that's part of the cytoskeleton of the cell. So I don't know exactly what his thought process was in picking these particular colors, but I mean, number one, they're kind of cool. They're bright, vibrant, fun to look at, and the use of color does help you distinguish one molecule from another. But beyond that, I'm not sure what other thought process he had. But really, you can make cells almost any color you want, but I do recommend trying to think about it, just like have some reason for your color choice, whether you think it's clearer for the audience to understand it or you just like it aesthetically. There's lots of different reasons you can give, for your color choices. All right, so that is it for today. I do want to let you know that we're going to have our second quiz next Monday. It's going to cover everything I talked about today. So light microscopy, electron microscopy, cryo-EM, cryo-ET, scale, know the difference between a nanometer and an angstrom and centimeter and all that kind of stuff. So we will do that, right at the beginning of class on Monday. And then this Wednesday, I've got another Horos exercise for you. So I'm all done. I'm going to stop the recording.